Gene expressions. Gene expressions can be represented through two main processes: transcription and translation. Let's begin on the basics. Gene is known as the functional part of DNA, or just a sequence of DNA. DNA, on the other hand, carries inherited genetic information used in the body's growth and system for its functions. The function of DNA is to code for protein, therefore gene codes for protein. Now, to understand how gene can be expressed, we will also need to understand the structure of DNA. DNA structure is made of many nucleotides. These nucleotides are then made up of a sugar carbon, a nitrogenous base, and a phosphate group. For DNA, there are 4 different types of nitrogenous bases: guanine, cytosine, adenine, and thymine. As a side note, RNA has almost the same nitrogenous bases as DNA, except that uracil replaces thymine. Moving on, one of these nucleotides are joined by another nucleotide through a bond between one of the sugar carbon and one of the phosphate group, known as the phosphodiester bond. All of this forms a polynucleotide. The sugar and phosphate units make up the backbone of this nucleic acid and showcases one strand of DNA. As one will probably know, DNA is double-stranded. This works due to hydrogen bonds existing between the nitrogenous bases. Just a reminder, hydrogen bonds formed between hydrogen and a more electronegative atom or group. They are weak individually, but collectively very strong. And now we have a double helix DNA structure ready to be transcripted into messenger RNA.